And this goes out to all of the people with a large sense of social justice. Right. Now your mother's going to listen to it and go, that's my son's voice in the background. Oh. Right. The coding regions are your genes, your structural genes. Your structural genes, which give you your proteins. Okay, so that's gene expression. Okay? The non-coding regions are everything else except for the structural genes. They're used a lot for the transcription factors and the control of gene expression. They also have large regions that have just been building up over time through evolution. Okay, and this is where we have our short tandem repeats, or we abbreviate that to STRs. Okay? These are areas short because there's not many nucleotides. All right, short, which is, we have that word oligonucleotides, which means a short sequence of 4 to 10 nucleotides. They can be various lengths. And they have repeating sequences. Okay? We'll just make one up. All right? Just say the STR we're dealing with is GATA. Now these increase in length due to errors in meiosis. Okay, so things can be duplicated and stay in there and not cause any problems. Because they don't cause any problems, they stay within the genome. They're a bit what we will term silent or neutral. So there would be one repeat. Right? If we had GATA, that would be 2 repeats. If we had 5 repeats, GA, 3, 4, 5. Thing to notice here is, as you have more repeats, the fragment length is longer. Okay, so because the fragment length of the nucleotides is longer, we can cut them out with restriction enzymes. Yep, and then we can run them through a gel to get a banding pattern called a DNA fingerprint. Now, what we'll do in just a second, we'll do one location. But when you have a DNA profile for biology, they'll do 20 locations. The more locations, the more specific the DNA profile or the DNA fingerprint becomes. And if you do 20 to 25 STR locations, then your profile will be yours. It'll be no one else in the world that has your DNA fingerprint. And that allows us to separate based upon non-coding DNA, whose DNA is who. Okay? So, any questions so far? So, it's used in your course, we use it for conservation. You want to be able to, we use it in conservation, we use it for artificial breeding, we use it for for fertilization, artificial fertilizations and things like that, but we don't want to have 2 related individuals breeding.
This is DNA profiling.
So we put the DNA into a well. Negative, yep. All right, positive end, yep. It's at one location, because one location you've got half the DNA from mum and half the DNA from dad, and you'll end up with 2 STRs for the one location. Okay, you can cut out that one location using restriction enzymes. Yep, because you know what is the flank, what we call a flanking sequence, which is what's before and what's after. You can cut it out. Yep. So we'll say here it gives us two bands. The one that is furthest along will be the shortest. So we might say that this is 5 and this will be 15. Okay? If we were doing a paternity case, right, you would want to know, you would get one from mum and one from dad, but you don't know which one has come from mum and which one has come from dad. So we do another DNA profile from your mother, right? And your mother's 2 STRs will be there and there. Okay? So we know that we've got a 15 and we know we've got, I don't know, an 8. Yes? And then the father's, which means that this STR here should come from dad. Yes? Because you have to get half from your mum and half from your dad. So the father's be there and there. Okay? And what you want to do is, if you are doing artificial breeding, the 2 individuals that you have selected in the breeding, you don't want them to have very similar DNA profiles, because that will mean that they are most likely related. You want to have ones that have the most genetic variability to make the next generation of offspring more able to cope with any changes in the environment. Okay? There have been questions in the past where they've found a cockatoo on the side of the road, you rehabilitate it, but there are 5 different cockatoo populations around the state, and to find out which one it belongs to, you do a DNA profile of the cockatoo, and then you do a DNA profile of all of the different populations, and the one that is most similar to, it's most likely going to come from that population. So it allows for the conservation. Yes?
If we did a DNA profile of everyone in the class, would they be dramatically different?
Yes.
Okay.
Unless, of course, there is someone that is related to another individual. Okay? If someone is related, even by a couple of generations or a couple of like cousins or something like that, they're still going to have similarities. Okay? So there's DNA profiling. It's used to identify an individual, but not through its genetic code. So you can look for similarities, and the more similar the DNA profile, the more related they are. All good?